The overall goal of this procedure is to quantify bacterial CD4 positive T-cell transinfection using a mouse cell model.
In vivo, some pathogenic bacteria, such as Listeria monocytogenes and Salmonella enteritidis, are able to infect T lymphocytes and modify their behavior. However, in vitro, T cells are not easy to infect.
Based on this observation, we investigated whether T cells can capture bacteria from infected dendritic cells during antigen presentation as occurs in viral infections.
We found that T cells capture bacteria and then kill them. Actually, we found that T cells do this more efficiently than professional phagocytes. These results contradict a central tenet of immunology, showing that cells from adaptive immunity can perform functions supposedly exclusive to innate immunity.
Today we present the protocol used to study the bacterial transinfection process in a mouse model.
Esteban, Veiga and I will be demonstrating this procedure along with Guillermo Ramírez-Santiago, Raquel, Garcia-Ferreras, 2 graduate students, and Mónica, Torres-Torresano, a technician.
Note that all the procedures described in this video should be carried out in the hood, using only sterile media, instruments, pipette tips, and culture dishes. In a non-tissue culture-treated sterile 15 cm Petri dish, add 1 microliter of 1 mg per milliliter lipopolysaccharide to the bone marrow-derived dendritic cells from a single mouse. Incubate the cells for 24 hours to increase major histocompatibility complex 2, or